Hey everyone, welcome back to a continuation of Lecture 3. This is part 2. This slide will pull together what autosomal dominant disease looks like across organ systems. On the left, you've got a table showing that autosomal dominant disorders don't stay in one lane. They actually affect the nervous, renal, GI, hematopoietic, skeletal, and metabolic systems. You'll notice familiar names like Huntington disease in the nervous system, polycystic kidney disease in the urinary system, familial polyposis coli in the GI tract, and hereditary spherocytosis and von Willebrand diagnosis in hematopoiesis. Marfan syndrome and osteogenesis imperfecta in the skeleton, familial hypercholesterolemia in metabolism, different systems, same inheritance logic. Um, Take a look at the diagram on the right. The pedigree style graphic on the right shows the classic vertical transmission pattern. Affected individuals in multiple generations with about 50% of offspring affected. When you see disease popping up generation after generation, autosomal dominant should be one of your first thoughts. Okay, now let's flip the script. Autosomal recessive disorders require homozygous expression, meaning both alleles at the same locus are mutated. So parents are typically carriers. They have one normal allele and one defective allele, and they're usually asymptomatic. When two carriers have a child, siblings have a 25% chance of being affected, a 50% chance of being carriers, and a 25% chance of inheriting two normal alleles. These disorders often present at birth or early in life, and they affect males and females equally. Most autosomal recessive disorders involve enzyme defects, which means loss of function, buildup of substrates, and metabolic consequences. Key takeaway here is autosomal recessive traits make up the largest category of Mendelian disorders, even though they're often less visible in family histories. All right, so inborn errors of metabolism are genetic disorders where the body is missing an enzyme, or the enzyme is present but doesn't work right. When that happens, a metabolic pathway gets stuck. And when a pathway gets stuck, one of two things usually happens. The substrate builds up behind the block and/or the product downstream is missing, which the body needed to function normally. Most of these are autosomal recessive, which makes sense because you usually need both alleles affected before you completely lose enough enzyme activity to cause disease. A big category inside inborn errors is glycogen storage diseases. These are disorders where a specific enzyme involved in glycogen breakdown, or sometimes glycogen processing, is defective. So instead of glycogen being stored and released normally, you can get abnormal glycogen accumulation, especially in organs like the liver and muscle. And depending on the specific disease, you can also see hypoglycemia, hepatomegaly, muscle weakness, or exercise intolerance. Sickle cell anemia is an autosomal recessive disorder caused by mutation in the beta globulin gene, or HBB. This is a classic missense point mutation where valine replaces glutamic acid at position 6 of the beta chain. That creates hemoglobin S, HbS. Here's the key concept. HbS behaves differently when oxygen is low. In low oxygen conditions, HbS polymerizes. It forms long, rigid chains inside the red blood cell. That's what makes the red blood cell stiff and sickle-shaped. And those sickled cells cause problems in two major ways. They break apart more easily, so hemolytic anemia, and they block small blood vessels, so vaso-occlusion and ischemic pain crisis. So when you hear sickle cell, think polymerization, sickling, hemolysis, and vessel blockage. And one more high-yield tie-in, people who are heterozygous with the sickle trait often have some protection against severe malaria. So it's a classic example of how a harmful allele can persist in certain populations due to a selective advantage. So now let's zooming out from the molecular change to the cellular and vascular consequences of sickle cell. When hemoglobin polymerizes, red blood cells become sticky, dense, and rigid. 3 qualities you never want in something that's supposed to flow smoothly. These misshapen cells can get stuck in small vessels, causing vascular obstruction, or break apart early, causing hemolysis. And that leads directly to chronic anemia and tissue ischemia. Normal red blood cells live about 90 to 120 days. Sickle cells, about 10 to 20 days. That's not a short vacation, that's a dramatically shortened career. On a peripheral smear, this is one of those diagnoses where you don't need a lot of imagination. Once you see sickled cells, you know what you're looking at. Cystic fibrosis is one of those diseases that shows up everywhere because the problem starts at the cellular level. The root cause is a mutation in the CFTR gene, which regulates chloride transport across epithelial cell membranes, especially in exocrine gland ducts. When chloride doesn't move properly, water doesn't follow it, and the result is thick, sticky mucus instead of nice, slippery secretions. In the lungs, that thick mucus traps bacteria, which explains the chronic lung infections. In the pancreas, ducts get blocked, enzymes can't reach the intestine, and nutrients don't get absorbed well. And in sweat glands, chloride can't be reabsorbed, which is why these patients have salty sweat. Fun fact, cystic fibrosis is diagnosed with a sweat test, and salty skin was historically noticed by parents who said their babies tasted salty when kissed. Medicine, romantic but weird. Cystic fibrosis is common among people of Northern European ancestry and was once almost universally fatal in childhood. Thanks to modern therapies, many patients now live into adulthood, which is an amazing example of how genetics and medicine evolve together. Phenylketonuria, or PKU, is a classic autosomal recessive metabolic disorder, and it's a perfect example of why newborn screenings save lives. In PKU, patients lack the enzyme phenylalanine hydroxylase, which normally breaks down phenylalanine. Without that enzyme, phenylalanine accumulates and excess phenylalanine is toxic to the developing brain. If untreated, PKU leads to severe intellectual disability, seizures, and behavior problems. But here's the good news. PKU is treatable with diet alone. By restricting phenylalanine early, meaning low protein diets and avoiding things like aspartame, children can develop normally. Worthless but fun knowledge, that warning label on diet soda that contains phenylalanine exists because of PKU. You may never need it, but someone absolutely does. PKU is the reason every newborn gets a heel stick test, even if they look perfectly healthy. This slide really highlights why recessive disorders can sneak through families unnoticed. In autosomal recessive inheritance, both parents are usually unaffected carriers. They don't have symptoms because one normal allele is enough to do the job. The disease only shows up when a child inherits 2 defective copies. Statistically, for each pregnancy, 25% of an affected child, 50% chance of a carrier, and 25% chance of an unaffected non-carrier child. And yes, this is where the Punnett squares earn their keep. The table shows that recessive diseases often affect metabolic pathways because enzymes are very unforgiving. They either work or they don't. So recessive diseases are often more common than dominant ones because carriers survive just fine and pass the gene along quietly for generations. Recessive genetics, the ultimate everything was fine until suddenly it wasn't situation. Now we're shifting gears to X-linked recessive inheritance, which explains why some diseases show up mostly in males. Here's the key concept. Males only have one X chromosome. If that X carries a defective gene, there's no backup. The disease is expressed. Females, on the other hand, usually have one normal X to compensate, so they're often carriers without symptoms. That's why carrier mothers have a 50% chance of passing the disorder to sons. Daughters may become carriers but usually don't show full disease. Also important, there's basically no such thing as Y-linked inheritance for disease. Most Y-linked mutations interfere with fertility, meaning they don't get passed on. Fun fact, X-linked diseases helped scientists understand inheritance before DNA was even discovered. Pedigrees walked so genomics could run. Hemophilia A is one of the most well-known X-linked recessive disorders, and it involves a deficiency of factor VIII, a key player in the clotting cascade. When factor VIII is missing or defective, clots don't form properly. This leads to spontaneous bleeding or bleeding that won't stop after even minor injuries. The most dangerous sites of bleeding include the brain, as in intracranial hemorrhage, the joints and bones, skin and soft tissue, internal organs like the spleen. Historically, hemophilia was called the royal disease because it spread through European royal families, including Queen Victoria's descendants. Hemophilia patients don't bleed faster, they just bleed longer. The faucet turns off very, very slowly, or not at all. Modern treatments with clotting factor replacement have dramatically improved life expectancy. Another win for molecular medicine. Duchenne muscular dystrophy is an X-linked recessive disorder, which is why it overwhelmingly affects males. If they inherit the mutation, there's no backup copy to save them. The underlying problem is a mutation in the dystrophin gene, which produces a protein that acts like a molecular shock absorber for muscle fibers. Without dystrophin, muscle cells are mechanically fragile. Every contraction causes microinjury. Over time, repeated damage leads to progressive degeneration of skeletal muscle, replacement with fat and connective tissue, and a steady loss of strength. This isn't limited to skeletal muscle. Cardiac and respiratory muscles are also affected, which is why heart failure and pulmonary complications ultimately drive morbidity and mortality. Clinically, patients lose ambulation in their early teens and become wheelchair dependent. Historically, survival into the early 20s was typical, though advances in cardiac and respiratory care are now extending life expectancy, which is something older textbooks often fail to mention. Duchenne is a textbook example of how structural proteins matter just as much as metabolic ones, and how muscle weakness can be a mechanical problem, not an energy problem. Fun but depressing fact, muscle cells in Duchenne patients are doing their job, they just can't survive it. On histology, think muscle fiber degeneration, replaced with fat and connective tissue and variability in fiber size. If there were ever a chromosome with a resume of bad decisions, it would be the X chromosome. This slide isn't here to make you memorize a list. It's here to help you recognize a pattern. The X chromosome carries a lot of genes, many of which are critical for normal function. When something goes wrong, there's often no backup plan, especially in males. Let's start with the musculoskeletal system. Duchenne muscular dystrophy lives here, and we've already talked about why losing dystrophin is catastrophic when there's no second copy to compensate. In the bloodstream, the X chromosome really shows off. Hemophilia A and B, classic clotting factor deficiencies. Chronic granulomatous disease, a problem with oxidative burst in phagocytes. G6PD deficiency, red cells that can't handle oxidative stress. Different pathways, same theme. One mutation, no safety net. The immune system is next. Agammaglobulinemia and Wiskott-Aldrich syndrome both highlight how X-linked defects can cripple immune function early in life. These patients don't just get sick, they get sick often and badly. Metabolically, diabetes insipidus and Lesch-Nyhan syndrome remind us that the X chromosome isn't just about structure, it's also deeply involved in regulation and metabolism. Leishmanian is, in particular, is one of those conditions you never forget once you truly understand the mechanism. And finally, the nervous system, Fragile X syndrome. It typically plays by non-Mendelian rules, which is why there's a little asterisk there, but it still disproportionately affects males for the same underlying reason. Gene dosage matters. If you're noticing that many of these disorders involve high energy systems, rapidly dividing cells, or tightly regulated processes, that's not a coincidence. The X chromosome is carrying a lot of biological responsibility. Fun fact, the X chromosome has over 1,000 genes, while the Y chromosome has fewer than 100. So when people say the Y chromosome is just there for sex determination, yeah, it is kind of freeloading. From a lab perspective, many of these diseases show up indirectly. So abnormal tissue architecture, unusual inflammatory patterns, or secondary organ damage. The slide might say blood or immune, but the downstream pathology often crosses systems. To now, most of what we've talked about with the X chromosomes has been recessive. One bad copy, no backup, game over. X-linked dominant disorders flip that idea on its head. Here, a single mutant allele on the X chromosome is enough to cause disease, even when a normal copy is present. These disorders are rare, and there's a good reason for that. When a dominant mutation sits on the X chromosome, it often causes severe effects in males and variable expressions in females, thanks to X inactivation. That combination is not evolution's favorite recipe. A key point to remember, affected fathers cannot pass these disorders to their sons because they give sons a Y chromosome. But all daughters of an affected father will inherit the mutant X. That inheritance pattern alone should make you pause and think when you see pedigrees. Two classic examples live on this slide. Vitamin D resistant rickets, where the problem isn't vitamin D intake but receptor signaling, and Alport syndrome, which affects type 4 collagen, meaning kidney, eye, and ear involvement, all tied back to basement membrane pathology. If you're seeing multi-system involvement, especially kidney, bone, hearing, and vision, and the pedigree looks weirdly skewed towards females with variable severity, start thinking X-linked dominant, not autosomal. X-linked dominant disorders are one of the few inheritance patterns where fathers can affect all daughters and 0 sons. It's like the genetic version of, I'm not mad at you, just disappointed. Alport syndrome is a great example of why understanding genetics matters in histology. When type 4 collagen is abnormal, basement membranes don't just look different, they function differently. If you've ever seen a kidney biopsy with a split or basket weave glomerular basement membrane, that's not random. This slide is where genetics stops being neat, clean, and satisfying. Polygenic disorders are difficult to determine because there isn't one gene to blame. No villain, no smoking gun, no tidy Punnett square. Instead, you're looking at the cumulative effect of multiple genes, each contributing a small amount of risk, often interacting with the environment. That's why inheritance patterns here look vague. You'll see clustering in families, but it won't behave like dominant or recessive inheritance. It's more like genetic peer pressure than genetic destiny. To make things even messier, similar clinical presentations can arise from non-genetic mechanisms: lifestyle, inflammation, epigenetic changes, aging, or even sheer bad luck. So when someone asks, is this genetic? The real answer is often yes, but also no. The examples on this slide should feel familiar: type 2 diabetes, hypertension, atherosclerosis, obesity, and many cancers. These are conditions where genes load the gun, but environment pulls the trigger, and sometimes repeatedly. If a disease is common, multifactorial, worsens with age, and shows up in families without a clear inheritance pattern, you're almost certainly in polygenic territory. Genome-wide association studies have identified hundreds of risk loci for type 2 diabetes, and yet none of them alone are sufficient to cause disease. So yes, science worked very hard to confirm that biology is complicated. Think about atherosclerosis. The plaque you see under the microscope didn't appear because of one bad gene. It's the endpoint of epithelial dysfunction, lipid metabolism, inflammation, and time. Histology shows you the receipt after genetics and environment finish negotiating. This slide explains why you only memorize a handful of chromosomal disorders, not because genetics is lazy, but because biology is ruthless. Most autosomal chromosomal abnormalities are incompatible with life. Full stop. When you gain or lose large chunks of autosomal DNA, you disrupt thousands of genes at once. Cells do not negotiate with that level of chaos. That's why chromosomal abnormalities account for about 1/3 of spontaneous abortions in the first trimester. These are not rare accidents, they're common outcomes of meiotic error. Most never make it far enough to get a name, a karyotype, or a board question. Sex chromosomes, however, play by slightly different rules. The X chromosome is gene-rich, but also partially buffered by X inactivation. The Y chromosome does not do much heavy lifting. As a result, sex chromosome disorders tend to be milder, and many individuals aren't diagnosed until puberty, when secondary sex characteristics fail to show up on schedule. So translation, autosomal aneuploidy usually ends development early, while sex chromosome abnormalities often slip through childhood unnoticed and announce themselves later, awkwardly, and hormonally. Humans tolerate sex chromosome imbalance better than almost any other chromosome imbalance, which is why Turner, Klinefelter, and XXX syndromes exist, but trisomy 8 adult syndrome does not. If you remember one autosomal chromosomal disorder for the rest of your life, this is it. Down syndrome is the most common chromosomal disorder compatible with life, and it is the leading genetic cause of severe intellectual disability. That alone tells you something important. Trisomy 21 is survivable in a way most autosomal trisomies are not. Chromosome 21 is small, but don't let that fool you, because gene dosage still matters. In the US, Down syndrome occurs roughly 1 in 1,000 live births, and about 95% of cases are due to meiotic non-disjunction, meaning the error happens before fertilization, not after. This is not a mitotic oops. This is a gamete level failure. Maternal age is the major risk factor, and the numbers are not subtle. A mother under 20 has about a 1 in 1,550 risk. By age 45, that risk jumps to 1 in 25. That dramatic increase reflects aging oocytes that have been sitting in meiotic arrest since fetal life. Time is not kind to spindle fibers. The classic karyotype is written as 47, X plus 21, where the X reminds you that the sex chromosomes are irrelevant to the diagnosis. It's the extra autosome that defines the condition. Male or female, the phenotype is driven by chromosome 21, gene overexpression. If you see a patient with congenital heart defects, characteristic facial features, and intellectual disability, and the karyotype reads 47 chromosomes, trisomy 21 should already be the leading diagnosis. Down syndrome is sometimes called the Goldilocks trisomy, not because it's mild, but because it's just compatible enough with life to be common. Trisomy 13 and 18 are not nearly as forgiving. All right, this slide is giving us the classic overview of Down syndrome, again, also known as trisomy 21. The key idea here is that there's extra genetic material, specifically an extra copy of chromosome 21, which leads to a predictable constellation of physical and clinical features. You'll notice the facial characteristics highlighted here: a flatter facial profile, oblique palpebral fissures, and epicanthic folds. These are often what prompt early clinical suspicion. But remember, a karyotype confirms the diagnosis, not appearance alone. Other common findings include a single transverse palmar crease, sometimes called a simian crease, hypotonia, and a gap between the first and second toes. From a pathology perspective, what really matters are the associated conditions, especially congenital heart defects, which occur in about 40% of patients. AV septal defects are the big ones to remember. There's also an increased risk of intestinal abnormalities like duodenal atresia and a predisposition to leukemia, particularly acute lymphoblastic leukemia. So yes, this is one of those conditions where genetics, embryology, and pathology all crash into each other at once. A fun but slightly depressing fact, chromosome 21 is one of the smallest chromosomes we have, yet causes one of the most recognizable syndromes. Genetics has a sense of iron. Now we're going to switch gears to Klinefelter syndrome, which is chromosomal disorder affecting phenotypic males. The most common karyotype you'll see is 47, XXY, accounting for about 90% of cases. This condition results from meiotic non-disjunction, meaning chromosomes didn't separate properly during gamete formation. The end result? Extra X chromosome material in someone with a Y chromosome. Clinically, Klinefelter syndrome is one of the most common causes of male hypogonadism. These individuals often have lower testosterone levels, which explains many of the physical findings we'll talk about on the next slide. You may see references to more feminine physical traits in older literature, but what we really mean is altered secondary sex characteristics due to hormonal imbalance. Language matters, especially in healthcare, so keep that distinction clear. Klinefelter syndrome is often underdiagnosed. Many individuals aren't diagnosed until adulthood, sometimes during fertility evaluations. So this isn't always something that's obvious early on. This slide fills in the clinical picture. Klinefelter syndrome is rarely diagnosed before puberty because many of the defining features don't become apparent until adolescence. Common clinical findings include long legs, relatively short trunks, and gynecomastia, which is breast tissue development due to altered estrogen to testosterone ratios. Testes are typically atrophic, and patients may have reduced facial and body hair, a higher pitched voice, and reduced muscle mass. There's also an increased risk of breast cancer, which is something people often forget because breast cancer is so strongly associated with cisgender women. Hormones matter more than labels here. Treatment usually involves testosterone replacement therapy, which can improve secondary sex characteristics, bone density, and the overall quality of life, but it does not reverse infertility in most cases. If you ever see tall male with infertility and gynecomastia, Klinefelter should start screaming in your head. Next up is Turner syndrome, which is essentially the chromosomal mirror image of Klinefelter. Instead of extra sex chromosome material, we're dealing with missing X chromosome material. The most common karyotype is 45, X, meaning there's only one X chromosome present. This condition affects phenotypic females and is the most common sex chromosome abnormality in females, occurring in about 1 in 2,000 live births. Turner syndrome is characterized by gonadal failure, leading to hypogonadism. Because ovarian development is impaired, many individuals experience primary amenorrhea and infertility. Fun fact, fun genetics reminder, one X chromosome is usually enough for survival, but not enough for normal ovarian development. Biology is rude like that. Amenorrhea simply means the absence of menstrual periods. At its core, menstruation happens when the ovaries produce hormones, estrogen and progesterone. Those hormones act on the endometrium, or the uterine lining. The lining builds up, then sheds if pregnancy doesn't occur. If any part of that system fails, menstruation doesn't happen. Think of it like tissue turnover. You already understand this concept from histology. No stimulus, no tissue response, no hormones, no endometrial proliferation, no endometrial buildup, there's nothing to shed. Nothing to shed means amenorrhea. So amenorrhea isn't the problem itself. It's a sign that something upstream isn't working. Menstruation never starts is primary amenorrhea. It's common in chromosomal disorders like Turner syndrome, usually due to non-functional or absent ovaries. So no estrogen, the endometrium never develops, therefore you have no periods. Secondary amenorrhea, menstruation starts normally, then stops. This can be caused by hormonal imbalance, pregnancy, stress, extreme weight loss or illness, and damage to reproductive organs. Hormone signaling gets interrupted, endometrium stops cycling. What's actually happening histologically? In amenorrhea, you would not see normal cyclic changes in the endometrium. So predictable phases, proliferative, secretory, and shedding. Instead, you might see thin, underdeveloped endometrium or an endometrium stuck in one phase due to hormone imbalance. In Turner syndrome, ovaries are replaced by fibrotic streak ovaries. No follicles, no estrogen. No estrogen, endometrium doesn't develop. Result, primary amenorrhea. This is why Turner syndrome is a leading cause of primary amenorrhea. So amenorrhea is not a uterine problem first. It's usually a hormone or ovary problem. If hormones don't talk to tissue, tissue doesn't respond. Histology logic applies beautifully here. A useless but interesting fact, the word amenorrhea comes from Greek meaning without, meno, month, and rhea, flow. So yes, it literally means no monthly flow. Medical terminology is aggressively literal. This slide highlights the physical and clinical findings you're most likely to see. One of the hallmark features in infancy is lymphedema, particularly of the hands, feet, and nape of the neck due to abnormal lymphatic drainage. As patients age, you may notice short stature, a low posterior hairline, and a broad chest with widely spaced nipples. From a pathology standpoint, the most critical complications are cardiovascular. So congenital heart defects, especially coarctation of the aorta, and bicuspid aortic valve are the leading causes of increased mortality in Turner syndrome. Ovaries are often described as streak ovaries, meaning they're replaced by fibrous tissue and lack functional follicles. Turner syndrome accounts for about 1/3 of all cases of primary amenorrhea. That's a pretty big number. One last reminder, individuals with Turner syndrome typically have normal intelligence, though there may be specific learning differences, especially with spatial processing. Up until now, we've been playing nicely within Mendelian sandbox, dominant, recessive, predictable ratios. This slide is where genetics politely asks Mendel to step aside. These are single gene disorders, but they do not follow classic Mendelian inheritance patterns, and that's exactly why they trip people up. Trinucleotide repeat mutations. Trinucleotide repeat mutations are all about unstable DNA. A short sequence, usually 3 bases like CAG, CGG, or CTG, gets repeated too many times. And the key concept here is anticipation. The disease gets worse and/or appears earlier in successive generations. The gene isn't just mutated, it's mutating harder over time. This is why family histories sometimes look deceptively mild until suddenly they don't. Some worthless but useful knowledge, the DNA polymerase isn't messing up, it's slipping, literally. These repeats form hairpin loops that confuse replication machinery. Genomic imprinting is where who you inherited the gene from matters. Same mutation, same chromosome, totally different outcome, depending on whether it came from mom or dad. That's because one parental copy is epigenetically silenced, typically via methylation, and the other is doing all the work. So if the active copy is defective, you get disease. If the silenced copy is defective, nothing happens, and that's what makes imprinting so sneaky. This is why Prader-Willi and Angelman syndromes involve the same chromosomal region, but present very differently. Mitochondrial gene mutations. Mitochondrial inheritance follows one brutally simple rule. If mom has it, everyone can get it. If dad has it, nobody does. Both sexes are affected, but only females transmit the mutation because sperm contribute essentially no mitochondria to the embryo. These diseases disproportionately affect high-energy tissues, so the brain, muscle, and heart, because mitochondria are the cell's power plants, and those tissues are energy hogs. Severity can vary within the same family due to heteroplasmy, different proportions of normal versus mutant mitochondria in different cells. So the big picture takeaway, these disorders aren't exceptions. They're reminders that biology is messier than rules. When inheritance patterns don't make sense, don't panic. Think mechanism, not memorization. Trionucleotide repeat mutations are defined by one key idea, amplification. A short 3-base pair DNA sequence gets copied too many times, and that expansion disrupts normal gene function. This is not a point mutation. Nothing is swapped or deleted. The DNA just won't stop repeating itself. The exact sequence varies by disease, CAG, CGG, CTG, but you'll notice a theme. Cytosine and guanine show up a lot. CG-rich regions from secondary structures like hairpins during replication, which makes them prone to slippage. DNA polymerase loses its place, reattaches incorrectly, and suddenly we have extra repeats. GC-rich regions stain differently and behave differently structurally. Sequence composition matters more than you think. Dynamic mutations equal anticipation. These mutations are described as dynamic, meaning the repeat length isn't stable. With each generation, the number of repeats can increase, and when that happens, the disease often shows up earlier, more severely, or both. This phenomenon is called anticipation, and it explains why a parent might be mildly affected while their child is profoundly impacted. Why this matters clinically. Repeat length correlates with severity, age of onset, and sometimes which tissues are affected. In other words, the mutation isn't just present, it's quantitative, and that number matters. Trinucleotide repeats were first recognized as a disease mechanism in the early 1990s, which means most of modern molecular genetics is younger than your instructor's career. Big takeaway, trinucleotide repeat disorders don't break Mendel's rules. They bend time. Same gene, same family, different outcomes. Because DNA replication isn't perfect and biology keeps receipts across generations. Fragile X syndrome is a classic trinucleotide repeat disorder. And if you remember nothing else, remember this. More repeats does not equal more protein. In Fragile X, more repeats actually mean gene silencing. The mutation is where mechanism matters. The problem lies in the FMR1 gene, which normally contains about 29 CGG repeats. In Fragile X, that number explodes, anywhere from 250 to over 4,000 repeats. At that point, the DNA becomes heavily methylated, transcription shuts down, and the gene is effectively turned off. No messenger RNA, no protein, no buffering of synaptic development. FMR1 produces a protein involved in synaptic plasticity, basically how neurons fine-tune communication. Without it, neural signaling becomes disorganized, which explains the cognitive, behavioral, and developmental features of Fragile X. This isn't neuron loss, it's bad wiring. Fragile X has a very recognizable phenotype, especially in males. Long, narrow face, prominent mandible, large, everted ears, macroorchidism, which becomes obvious after puberty. Fragile X is the most common inherited cause of intellectual disability in males, second only to Down syndrome overall. The term fragile comes from early karyotyping studies where the X chromosome appeared to break under folate deficient cultural conditions. It's not actually fragile, it's just epigenetically muted and deeply misunderstood. Big takeaway is Fragile X shows us that too much repetition can be just as harmful as a missing gene. It's not mutation by damage, it's mutation by excess and silence. Gene imprinting is where genetics stops being fair and starts keeping receipts. You inherit 2 copies of every autosomal gene, one from mom, one from dad. But here's the twist. Sometimes only one of them is allowed to talk. Roughly 200 to 600 genes in the human genome are imprinted, meaning one allele is epigenetically silenced. Not mutated, not deleted, just muted. And which allele is silenced depends entirely on who it came from. The terminology is simple but easy to overthink. Maternal imprinting, the maternal allele is silenced. Paternal imprinting, the paternal allele is silenced. So if the active copy is damaged or missing, congratulations. You now have a disorder caused by perfectly normal DNA that just isn't being expressed. Imprinting doesn't happen after fertilization. It happens before, during gametogenesis. The egg and sperm arrive pre-labeled with epigenetic sticky notes saying, use this one or ignore this one. Once fertilization happens, those labels are copied into every somatic cell via mitosis. You don't get a redo. During the formation of your gametes later in life, those imprinting marks are erased and reset, not inherited directly, which means your genome remembers your parents, but your gametes only care about you. Biology is equal parts elegant and petty. So a big takeaway in genomic imprinting, the same genetic deletion can cause completely different diseases depending on whether it came from mom or dad. The DNA sequence is identical. The parental origin is what matters. This slide is genetics being dramatic on purpose. Same chromosome, same region, same deletion, completely different diseases, depending on which parent it came from. The setup is chromosome 15q11 through q13. Both Prader-Willi and Angelman syndromes involve a deletion in 15q11-q13. Nothing exotic, no fancy rearrangements, just missing DNA. The trick is that this region is imprinted, so one parent's copy is already silenced. Lose the active copy? That's where the trouble starts. Prader-Willi syndrome, think hunger and hypotonia. Prader-Willi syndrome occurs when there is maternal imprinting, so meaning the maternal allele is silenced, deletion of the paternal allele, and so translation, the only copy that should be active is gone. Clinical patterns to recognize is hypotonia, often severe in infancy, intellectual disability, hyperphagia, obesity if not strictly controlled, short stature, small hands and feet, and hypogonadism. Important to note are these patients don't just overeat, they lack normal signaling to tell them that they're full. This is hypothalamic dysfunction wearing a genetic name tag. Angelman syndrome, think ataxia and laughter. Angelman syndrome flips the script. Paternal imprinting, the paternal allele is silenced. Deletion of the maternal allele. Same chromosomal region, different parent, different outcome. So classic features of Angelman syndrome is severe intellectual disability, ataxic gait, seizures, and inappropriate or frequent laughter. Angelman is sometimes nicknamed the happy puppet syndrome. Outdated terminology, but the ataxia and laughter combo is unforgettable. So if you see obesity and hypotonia, think Prader-Willi. If you see ataxia, seizures, and laughter, think Angelman's. Same deletion, different parent. Ataxia, for those that don't know, is loss of coordinated movement due to dysfunction of the cerebellum or its neural pathways, not muscle weakness. So these are the facial and physical features, what we're supposed to notice. So on the left, you see a composite illustration. On the right, a real patient. Common facial features you may hear described are narrow bifrontal diameter, almond-shaped eyes, the thin upper lip, downturned mouth. None of these alone are diagnostic, but together with the right history, they should make you suspicious. Think of these as context clues, not answers. Remember what drives Prader-Willi. So loss of paternally expressed genes on chromosome 15, a hypothalamic dysfunction, and that explains everything else. So hypotonia in infancy, poor feeding early, which is uncontrolled hyperphagia later, obesity that is not behavioral, short stature, and hypogonadism. This is not a lack of willpower disorder. It is a broken satiety circuit. Many patients with Prader-Willi will require locked food environments, literally. This is one of the few conditions where food access has to be treated like medication. Dose controlled, timing controlled, access restricted. That's not parenting style, that's pathophysiology. Hyperphagia means abnormally increased appetite and excessive eating. It is usually caused by dysregulation of the hypothalamus, the brain's appetite control center. The body does not receive or respond to I'm full signals, which results in rapid weight gain if not managed. So the histotech way to think of it is the hunger on switch is stuck and the off switch doesn't work. Mild strabismus. Strabismus is misalignment of the eyes. They do not point in the same direction. Mild means it may be subtle or intermittent, caused by poor coordination of extraocular muscles or their innervation. This can affect both perception and visual tracking. Histotech the muscles are present, but the timing and signaling between them is off. Hypogonadism. Hypogonadism is reduced function of the gonads, ovaries or testes. Leads to low sex hormone production, results in delayed or absent puberty, infertility, and reduced secondary sex characteristics. The organ is there, but hormone output is insufficient or absent. This slide is the mirror image of Prader-Willi. Same chromosome, same region, completely different neurologic phenotype. Angelman syndrome results from loss of maternally expressed genes on chromosome 15, specifically involving the UBE3A gene. In neurons, the paternal copy is normally silenced. So if the maternal copy is gone, there is no backup. Same deletion, different parent, entirely different disease. Angelman is not similar, does not equal Prader-Willi. Angelman syndrome is not subtle. Hallmark features include severe intellectual disability, ataxic jerky gait, seizures, minimal or absent speech, inappropriate laughter and smiling. That constant smiling you see here, that's not happy demeanor, that's a neurological finding. Facial features, you may notice wide mouth, prominent smile, wildly spaced teeth, prominent lower lip, and light hair or eyes in some patients. Again, faces don't diagnose genetics. Angelman patients often look alert and joyful despite profound neurological impairment. Welcome to the only time in genetics where dad is completely irrelevant. Mitochondria have their own DNA, and it is inherited exclusively from the mother. Sperm bring DNA, eggs bring DNA and mitochondria. Dad's mitochondria, destroyed after fertilization like they never existed. This is why mitochondria inheritance is sometimes called maternal-only inheritance. Not sex-linked, not autosomal, just maternal. Mutations in mitochondria DNA impair oxidative phosphorylation. Translation, less ATP, less endurance, more symptoms when energy demand increases. These patients often look fine until they're asked to do something. Mitochondria diseases hit tissues that cannot afford an energy shortage. So skeletal muscle, cardiac muscle, brain, and the peripheral nerves. If a tissue relies heavily on aerobic metabolism, it's on the hit list. Red flags include exercise intolerance, muscle weakness, neurologic symptoms, seizures, vision or hearing loss, and symptoms that worsen with stress, illness, or exertion. Some worthless but useful knowledge. Mitochondria DNA mutates faster than nuclear DNA because no histone production, limited DNA repair mechanisms, and constant exposure to reactive oxygen species. So basically, mitochondria live dangerously. All right, take a breath. You've officially survived chromosomal abnormalities, imprinting, trinucleotide repeats, and mitochondria. That's a lot of genetics in a short amount of time, and hopefully you're still here. We've got one more lecture left for this week, and then you get a break from new content. Use it to review, regroup, and let your brain catch up with your notes. If things feel a little scrambled right now, that's normal. Genetics always feels messy before it clicks.